everyone. I hope you're enjoying the Big Data Health Science Center annual conference thus far. I'd like to take a moment before the next session to introduce our pilot project program. You might have heard about it already, but this is just a little more information to prepare you for your own applications to submit. So this program, we released our RFP on January 11th this year. The applications are due April 15th, and we will notify applicants about the outcome of the process in late June of this year. Projects will start in August of 2021. The purpose of this program is to support projects that can leverage existing data to address critical issues related to health behavior, clinical care, healthcare delivery, and population health. We want to promote interdisciplinary collaborations in big data health science research and support researchers who are endeavoring to learn new skills and apply them in their respective fields to transform the way healthcare is delivered and improve population health. So let's go ahead and introduce the speakers for this session. All of the speakers are previous winners of the Pilot Projects program from last year and will be presenting some of their important work using big data analytics for population health research. Our first researcher is Dr. Wan Wang, who is an assistant professor of biostatistics in the University of South Carolina. Her current research is focused on topological learning of multimodal neuroimaging data with applications to clinical diagnosis and prognosis of epilepsy and post-stroke aphasia. The title of her presentation is Topological Network Analysis and Graph-Based Deep Learning of Multimodal MRI, an Enigma Epilepsy Study. The next speaker is Dr. Kai. Dr. Kai's research focuses on the development and application of bioinformatics. He uses statistical and computational methods for analyzing genomic data to address public health issues. His research aims to develop efficient methods for sequencing data analysis, such as differential expression analysis, batch effect correction, and single-cell RNA sequencing data analysis. He also uses machine learning methods and computational toxicology methods in his research. The title of his presentation today is Big Genomic Data Analysis to Characterize TME Methylation Expression, Regulatory Axis in Colorectal Tumors. And our final speaker for this session is Dr. Hongmei Balafar. Dr. Balafar's research activities fall into two broad categories of research: computational medicine and computational biology. The computational medicine tier of his research is focused on development of computational modeling of human response to different medical treatments or methods of rehab. His group has demonstrated success in relation to administration of different methods for alleviation of symptoms associated with sickle cell anemia. And the second tier of his research focuses on the problem of protein folding. Dr. Balafar's current research is funded by NIH and NSF and focused on exploration of hybrid approaches that combines minimum experimental data with novel computational approaches. The title of his presentation today is Utilization of Artificial Intelligence-Assisted Data Analytics to better predict progression of chronic kidney disease to end-stage than currently available predictive markers. We're looking forward to hearing you all today. And welcome again. I hope you're enjoying your time at the conference.
Hello there. My name is Yuan Wang. I'm an assistant professor of biostatistics at the Arnold School of Public Health of the University of South Carolina. I will be presenting an outline of a project funded by the Big Data Health Science Center Pilot Project Program in 2020. The project is motivated by the Enigma Epilepsy Initiative for improving diagnosis and prognosis of epilepsy through a big neuroimaging data approach. So, what is epilepsy? Epilepsy is a severe neurological disorder marked by sudden recurrent episodes of sensory disturbance, loss of consciousness, or convulsions. Epilepsy affects 65 million people around the world, and 3.4 million people in the United States. According to the Epilepsy Foundation statistics, more people live with epilepsy than autism spectrum disorders, Parkinson's disease, multiple sclerosis, and cerebral palsy combined. Epilepsy is primarily treated through anti-epileptic drugs, which are effective in approximately 70% of the cases. The rest are classified as drug-resistant or refractory epilepsy. When a patient fails 2 or more anti-epileptic drugs, they are typically referred for epilepsy surgery. A standard epilepsy surgery removes a part of the brain called the epileptogenic zone, which accounts for the neuronal network capable of generating seizures. Pre-surgical assessment of post-surgical seizure freedom is therefore critical. There are 2 main diagnostic tools to localize the epileptogenic zone for surgical resection: electroencephalography or EEG and magnetic resonance imaging or MRI. Since EEG requires considerable expertise of the specialist reading the signals, MRI has become a critical tool in the evaluation of drug-resistant epilepsy patients undergoing pre-surgical assessment. It can help identify lesions associated with a seizure focus, thus improving the odds of post-surgical seizure freedom. However, some patients do not show visible lesions on MRI. For instance, between 20 and 40% of focal epilepsy cases do not show visible lesions on MRI. And generalized epilepsy is by definition non-lesional. It is therefore important to quantify brain structural patterns in patients achieving favorable surgical outcomes. It will have a sustained impact on epilepsy care by improving patients' cognition, quality of life, psychosocial function, medical costs, and most importantly, reducing mortality rates. Existing studies have already shown promising results on the data-driven approach to structural and diffusion MRI, or SMRI and DMRI for short, in predicting drug response and surgical outcomes across epilepsy syndromes. And the network modeling approach to the SMRI and DMRI appears particularly promising. Taking a big neuroimaging data approach, the Enhancing Neuroimaging Genetics through Meta-Analysis Consortium, or ENIGMA, is now the largest international study of the human brain using neuroimaging. ENIGMA's Global Alliance for Worldwide Imaging in Epilepsy, or ENIGMA Epilepsy, integrates MRI data from over 2,100 epilepsy patients from 24 sites in 14 countries. ENIGMA Epilepsy provides unprecedented power of analysis and a unique opportunity to answer complex clinical questions in epilepsy. ENIGMA Epilepsy motivated this pilot project. In this project, we have 2 aims. Under the first aim of learning brain connectivity across epilepsy syndromes, our goal is to detect subtle structural brain abnormalities associated with both focal and generalized epilepsy syndromes with topological network analysis of the ENIGMA Epilepsy SMRI and DMRI data. Under the second aim of predicting treatment outcomes across epilepsy syndromes, our goal is to predict epilepsy treatment outcomes by building graph-based deep learning algorithms on the Enigma Epilepsy SMRI and DMRI data. In our first aim of learning brain connectivity across epilepsy syndromes, we take the approach of first building node-based and edge-based correlation network models on the Enigma Epilepsy SMRI and DMRI measurements and obtain their corresponding network filtrations to measure the strength of connections between brain regions through their persistence in the filtrations. We then extract persistent network features and combine them with clinical variables for group analysis to delineate brain connectivity patterns associated with different epilepsy syndromes. On the right, we will see an illustration of the approach. To begin with, the structural connectivity of the brain network is represented by correlations between the brain regions of interest. The correlations are typically thresholded to reveal significant connections in the network. But instead of focusing on a subnetwork based on a single threshold, which could be arbitrary, we take the network filtration approach to track connections or features formed by the connections that persist through the network as we thresholded with a range of values. Once we obtain a network filtration or a group of network filtrations for one epilepsy syndrome, then we can compare the persistent features of the network filtrations across multiple syndromes. In the second aim of predicting treatment outcomes across epilepsy syndromes, we will train graph-based deep learning algorithms on the Enigma Epilepsy SMRI and DMRI data to predict the outcomes of anti-epileptic drug and surgical treatments. We will use state-of-the-art pooling techniques such as diff pool illustrated on the top right for brain network classification. We will also investigate ways to boost the prediction accuracy of treatment outcomes by refining existing approaches with novel topological embeddings. I will serve as the principal investigator of the project, with input from two clinical consultants, Dr. Carrie McDonald of UC San Diego, who is also the co-chair of Enigma Epilepsy, and Dr. Leonardo Bonilla of MUSC, who is a highly experienced epileptologist and a site PI on Enigma Epilepsy. We will work together to ensure the clinical relevance of results in the project. The projected timeline for Aim 1 of the project is March through August this year, and Aim 2 is expected to be completed by February next year. Here are the references cited in the talk. Thank you for your attention.
Hey everyone, this is Guoshui Kai from the Department of Environmental Health Science in the School of Public Health in the University of South Carolina. So today I'm happy to talk about our pilot study, the big genomics data analysis to characterize tumor microenvironment and methylation expression regulatory axis in colorectal cancers. This study is collaborated with Dr. Xiao from the Department of Epidemiology and Biostatistics and Dr. Nagarkati from the Department of Pathology, Microbiology, and Immunology. We focus on the colorectal cancer because it's the third leading cause of the cancer-related deaths in the US. In 2019, the number of CRC-related deaths in US is above 50,000. The number is pretty high, especially it gives the high risk to the patients, especially for those who with the advanced stage. One major factor for the CRC progression for this issue is the CRC resistance to the treatments. And we know the tumor phenotypes and the resistance to the treatments, they are related to many factors. So they are related to the tumor intrinsic features, such as the tumor genetics, like the mutations in TP53 and other key genes. Emerging studies, now we know more, and emerging studies show that so those phenotypes of CRC, they are not only related to the tumor intrinsic feature, but also related to or depend on tumor intrinsic microenvironmental factors. So it depends on factors in their surrounding environments. We call this tumor microenvironments, or TME. But we don't know how the TME, the microenvironments, interact with the tumor cells. But we don't know how, but we know it's very important because tumor need to escape from the attack of the immune systems in their microenvironments. And also the microenvironments can provide favors environments to the tumors, to the tumor progression, like the chronic inflammation. And the study has shown that the epigenetic mechanisms such as the methylation play important roles in regulating the tumor cells in the tumor microenvironments and the tumor cell interactions. So it's give us a very important clues, and it's asked for a genome-wide investigations on TME and the methylation and the expression regulatory axis. But however, there's no genome-wide investigation about that has been performed because it's very difficult and it's challenging. Because previously we don't have the data, we don't have the multi-level data, and we don't have a good integrative methods. So now we have a TCGA project with excellence, and which collected large-scale tumor samples and as well as measure their genomics and collected their clinical features. Also, there are established methods. The method established for integrative data analysis, including ours novel methods for integration of a methylation and gene expression. So we have large-scale data right now. We have methods as well. So these are nothing I need to mention that the tumor microenvironments, they are tissue specific. So for breast cancer, they have breast cancer specific TMEs. For CRC, they have a CRC tumor specific tissue specific TMEs. So in all this, it's very hard to study those tumor tissue specific TMEs using cell lines and using as a reference. But now we have the single cell data, single cell study. It's give us a very valuable opportunity to study this issue, these problems. This give the single cell study give us a single cell measurements of transcriptomics, and they give us the profiles, they give us the compositions in the TMEs. Okay, so the long-term goal of the project is to identify the key factors in the interaction of TME and the tumor cells. And we expect to identify the potential new targets for improved treatments. And we also expected to understand the interaction of tumor microenvironments and the tumor cells. Okay, so in the pilot study, our first aim is to identify the variation of the TME cell composition in CRC and its mutation drivers and clinical effects. And the second aim is to study the genome-wide aberration of transcriptional regulatory methylation in CRC and its potential mutation drivers, underlying pathways, and clinical effects. So the first aim is for TME self-composition, and the second aim is for CRC tumor cell intrinsic features. And in M3, we integrate them. We want to investigate the interaction between the TME cell composition, methylation, and expression in CRC. So it's a very comprehensive analysis, and we need the support from multi-level data, including the CRC tumor genomic data sets from 460 TCGA samples, including the RNA-seq transcriptomics, the 450K DNA methylation, the whole genome data, and the whole genome SNP data, and the whole exome sequencing, somatic mutation calling, and of course the important clinical data. And again, we want to study the tumor specific TME, so we analyze the CRE tumor single cell RNA-seq data from 11 colorectal tumors, and we want to use the data as a reference to infer the TME composition in those 460 TCGA samples. So our study design is shown here in this figure, and we will work on a multi-level data, and we will use multiple tools, including established tools by others and our tools, developed tools, for example, the multicellular tool for CNV calling, which is designed and developed by Dr. Xiao's group. Dr. Xiao is expertise in the CNV identification, CNV calling from whole genome SNP data and the sequencing data. And we established the pipeline for bulk tissue RNA-seq data analysis and the single cell data analysis and the sequencing data analysis, including, with multiple methods integrated into the pipeline, including our newly developed SDC for reducing the noise from the single cell data and the steps for deconvolution analysis, and our integrative methods for methylation and gene expression to dichotomize the methylation. So we will obtain multiple variables, including CRC-specific TME cell composition, and the CRC-specific expression, regulatory methylation, and the dichotomized methylation status. And also we have mutation data, we have a copy number variation data, and we have clinical information, including age, gender, tumor stage, and survival informations. So, and further, we will use appropriate statistical methods, including the generalized linear model, survival analysis, clustering, gene set enrichments, mediation analysis to test the association to study those three aims. Okay, in the current phase, we download the data and we process the data, and it takes a lot of efforts, and we develop the platform, the web server to hosting, analyzing, visualizing those data sets, omics, large-scale omics data, and single-cell transcriptional data. So we develop scanner, which is a single-cell RNA-seq annotated viewer for single-cell data annotation and visualization. We have analyzed specifically for this study, pilot study, we have analyzed and annotated and visualized the data from this 11 colorectal tumors. As shown in this figure, we identified the cell cell types, and we construct the profiles for each cancer types as a reference for the TME composition deconvolution from the bulk tissues. And the second platform is Caesar, is a single cell inferred site-specific omics resource for tumor microenvironments. So it's a novel web server resource for association study between tumor-specific TME computation and the tumor omics in various cancer, including the CRC. And here is so it's a pretty comprehensive. And this figure, just to show an example, we identified the top associated genes with the TME, CRC TME. And we showed their associations in the heat map. Okay, and you can go into those websites. It's totally open and it's open source, and we have a lot of data inside, and you can play with. And if you have other interested data sets, you can contact me and send an email, and we can work on this together. And if you have a private data, we have a module with the password protected module. You can have the account, you can have the password, and you can have the private data. But you can also use all the resources and the visualization modules in our tools. Okay, so it will promote the biomedical analysis. I think an example, I will show you an application with scanner in the COVID-19 research. We analyzed the normal people's branchial epithelial cells, and from smokers and non-smokers, we found they have a different composition. The smokers largely lose the club cells, but excessively produce the goblet cells. And the ACE2, we know ACE2 is the receptor of the COVID-19, of the SARS-CoV-2 virus, and the smokers have ACE2 expressed in their goblet cells, but non-smokers have expression in their club cells. So the smoking remodels branchial epithelial cells and affects the ACE2 gene expression, and this remodeling could contribute to the disparity in the infection susceptibility, disease severity, and treatment outcome, between the smokers and non-smokers. And further, we analyzed the data from the lungs and the blood of the people who got COVID-19, and we found the smokings and regulates the expression of its ACE2 and another receptor, CD209 in lungs. And SARS-CoV-2 modulates the dendritic cells differently in lungs and in blood. And the severe, this red ones, severe COVID-19 cases have a weak dendritic cells, less capable of modulation and immune initiation. We know these dendritic cells are important, the immune cells who is representing the antigens to T cells and activates the immune system. So if it loses the power, your immune system cannot be activated. So that could be a reason for the severe COVID-19. Okay, so I would think Dr. Xiao, Dr. Nagakati's, Chris Amers from Baylor, and David Christiani from Harvard, Michael Whitfield from Dartmouth, and Johan from Laval University. So I would thank for the efforts on the COVID-19 research. And if you so we published the paper in Medicov and the AJRCC and the current one is also published in the Medicov. So if you are interested, you can just go online to find the paper. They are open source, the open access. And if you have ideas and thoughts and you have questions, just send me an email and I would be more than happy to answer your questions and to develop ideas and the thoughts.
Everyone, my name is Hamid Valafar, and with the help of Dr. Matthew and Brendan today, we are going to give you a brief summary of where our pilot project funded by Big Data Health Science Center is. Our focus is in trying to develop an AI-assisted data analytics to better predict the progression of chronic kidney disease. Our approach is going to be, the first half of this talk is going to be conducted by Dr. Matthew, where he's going to give you a little bit of background on CKD, chronic kidney disease, and motivate the need or basically the niche for where AI can fit. And then I am going to take the second half of it, where I am going to discuss some of the AI that, some of the examples of AI we have utilized in the domain of healthcare in my lab.
Okay, thank you, Dr. Valafar, and I want to also extend my thanks to the BDHSC folks for giving us the opportunity, first for the grant and also for the opportunity to present this project today. So I'm going to give a little background about why chronic kidney disease and what the specific problems will be related to how we're going to use the expertise from Dr. Valafar's lab to be able to answer the questions regarding chronic kidney disease. So first, a little scope about the problem of chronic kidney disease, and this is primarily data from the United States. So as end of 2016, there were greater than 700,000 persons with end stage of chronic kidney disease requiring renal replacement therapy. And I'm going to refer to this as chronic kidney disease, or CKD stage G5D. That's G5 is the fifth stage, and D refers to dialysis, and I'll quickly overview this later. An annual incidence of this stage of chronic kidney disease in the United States is reported to be about 373 per million persons per year. And this is reported from the United States Renal Data System, which is the national subset of the Medicare that supports dialysis payments in the United States. Now, despite a relatively low prevalence, about 0.2% of the population, CKD G5D is associated with very high morbidity and mortality. In addition, the cost of care for CKD G5D are astronomical. From this United States Renal Data System report, we see that between 2015 and 2016, the Medicare fee-for-service expenditure rose from $33.8 billion with a B to $35.4 billion. That's a 4.6% increase. And to give a little perspective, this one disease state, this is the end stage of a larger problem of chronic kidney disease. And this one disease stage consumes about 7% of the total Medicare budget. So this is an incredibly important problem, not only for patients, but also for the payer system of the healthcare system. And in fact, it was considered such an important problem that the prior executive administration, the president administration, chose this as one of the areas to target for improvements in health expenditure to reduce the total costs that are needed and improve quality of care for patients. So a little bit more about how we get to the point of end stage, since that seems to be the biggest problem. So as you saw, I spoke a little bit about the stages. So chronic kidney disease doesn't just happen. For the most part, we don't end up, the majority of patients don't just end up with end-stage kidney disease or CKD-G5D. They usually start at earlier stages of disease. And here depicts how renal function just slowly declines until you get to the lowest stage of kidney disease prior to needing end-stage kidney disease. And so what we have really thought as to be the gold standard in the care of chronic kidney disease is how do we target disease here to better prevent patients ending up down here with nearly no kidney function left. Currently, the best treatment that we have for end-stage kidney disease is renal transplantation. And that provides the greatest quality of life as compared to hemodialysis, peritoneal dialysis. However, as we all know, there's very few organs available, even though we have 2 kidneys compared to a heart and liver. But it's still a very limited resource, and not everyone qualifies for renal transplantation. So this is still not an answer for every patient who has end-stage kidney disease. And so there really is a lot of room for improvement here. So how do we identify patients with chronic kidney disease? So chronic kidney disease at its essence is defined by the level of functioning of the kidney. And we estimate that by looking at a value called the glomerular filtration rate. And that can be simply looked at as the working of the individual unit of function within each of the kidneys. And the glomerular filtration is essentially how the kidney filters the blood at every little filter unit, and we estimate the total sum of that unit. Now, we don't directly measure this. That's why it's called estimated in most human settings and clinical settings. And so we have to be able to estimate this. And how we do this is there's a measured variable called the serum creatinine, which is basically a breakdown product. It's a waste product, and it's cleared by the kidney. And so the level of that estimates the level of functioning of the kidney. Now, the estimating equation also uses several other variables, including age, gender, and race, plus the serum creatinine. And what I have listed for you here is the most current version of the formula that is used to estimate this. And this is the serum creatinine, female, gender, and Black race. And this is the citation for that. So the primary target of identifying and treating patients with this earlier stages of chronic kidney disease, as we mentioned, is really to try to prevent the progression to CKD G5D. Now, the challenge with this, even though we spoke about the expense and the problem of CKD G5D, and we mentioned this briefly, is that very few patients actually progress to CKD G5D. And we saw that the prevalence is actually fairly low in the population, and even in the United States population. As we look here, we look at this left-hand column here, the rate per 100 persons per year is about 0.5, depending, and it's really low, depending, and it's even lower depending on where you're starting in terms of the kidney function. So if you're starting at higher kidney function, greater than 60 mLs per minute, or we can just assume greater than 60% kidney function, your rate of progression is 0.5% per year. In comparison, and this is an older cohort where this data was derived from, but it's still similar even if you look at younger cohorts. But if you look at this cohort, if you look at their respective risk of death, their rate of death per year is 6.8 per 100 per year. So you're talking about 0.5% per year progressing to end-stage renal disease or CKD stage 5, and about 6 to 7% per year dying. So identifying patients, obviously these are two separate problems, and we're right now focusing on the end-stage renal disease problem. But identifying these patients is really important and really a challenge because there is so few that actually progress. And this is data, a much larger data set that looked at this same problem. about actual progression of chronic kidney disease. And this is a global cohort of patients, about 700,000 patients, and looking at actual rates of progression of patients over time and where they start from in their chronic kidney disease. And as you can see, the higher you are, typically you don't progress very much over time, and the lower you are, there's more significant progression over time. But even within this lower cohort, if you look at this is, I apologize, this is not expanded, but even if you just get a general look at this graph here, there's an incredible range of patients. There are some patients that have a fairly flat progression, and then there are others that truly do progress over time. So we're not dealing with a truly homogeneous population where you just look at this one value of GFR and you can say that defines what's going to happen to you over time. So we need better prediction to be able to know, well, who really is going to target and what are the ways to really target those patients to better apply therapeutics that are available. So currently, what we use to predict who will progress to end-stage kidney disease is the kidney function, as we talked about, eGFR. We still use age because even if age is in the formula to predict, to estimate the glomerular filtration rate, a glomerular filtration rate in someone who's older and younger still means something very different. The risk for progression is very different. So it still matters. And again, gender is still an important variable. And this is a reference where they had created a formula based on these large cohorts to look at, can we predict based on some of the variables that we have, who is at highest risk for progression? And using standard logistic regression and linear regression parameters, the best available prediction that we have right now is age, gender, the kidney function, and a value called the ACR, which is the amount of protein that's spilling in the urine. And this performed just as well, if not better, than when you could simply lump in some more clinical variables within a very standard prediction algorithms that are using standard statistical methods to be able to do this. And we can see this within the C statistics that are reported. And the 4 variable equation versus the 8 variable, at 3 years, they were pretty comparable. And at 5 years, the 4 variable performed just slightly better. So using our current available methods, we only have prediction available based on just the kidney function and the albumin, despite the fact that we have many variables that we measure routinely on these patients. And using standard statistics, we cannot improve beyond these variables, even when you add other variables. So that has been an incredible limitation. Now, even though these are fairly good C statistics, almost close to 90%, we're still talking about a very bad endpoint. And so if we can even improve on this with the available clinical variables that we have, we'd be able to even pull back on even the 10% that we're missing. So with that background, we decided that we would look at some of the limitations that we had. There's limited availability of variables that were used to analyze in many of these cohorts. And even though the prior cohort, as we can see, used some more clinical variables, there's still far more clinical variables that we collect sometimes on a monthly basis, if not at least on a quarterly basis on these patients, that we simply just discard, that we don't even look at. Limited number of participants. In many of these studies, thousands or a few thousands is the most that you can have. The previously one had close to about 700,000 patients, but again, limited in terms of the analytics that were used and the variables that were available to them. And then again, as we spoke, limitation of the traditional risk prediction models. So what we proposed was to use now some of the more modern techniques and machine learning algorithms. in conjunction with a very comprehensive and robust database in the VA data set to see if we can improve on the prediction ability at various stages of early stage chronic kidney disease of who will progress. So what has been done previously? So Xiao et al. Has looked at this and looking at trying to use more clinical data points to be able to predict. And as you can see, they were able to improve on the prediction curves compared to just using albuminuria or kidney function alone with a pretty reasonable area under the curve. But what are the problems with this data set that they were using? It's a very small cohort, 551 patients, and they tried to use algorithms that were optimized for small data sets. But it's also a very limited racial cohort. This was a completely Asian cohort, and they also used more surrogate outcomes rather than hard outcomes. They used the progression of the protein in the urine, which is a fairly good prediction, but there's incredible range in terms of patients who will actually progress. So what we plan to do is try to use more hard endpoints, larger data sets, and see if we can even improve on these in a more larger representative data set for the United States as well. So what have we been able to accomplish? So within the last 3 months, we were able to get data approval to be able to access the VA data set. And in the same amount of time, we were able to get Brendan credentialed within the VA data, within the VA system so that he can start accessing it. And within a very short period of time, Brendan really did a great job looking at the data set and has looked at what we have available. And as you can see in this simple, very starting preliminary flow diagram, the amount of data that's available for us to work with. Number of variables that were recorded amounted to about 205 million variable points, and number of just kidney function estimated variables about 156 million, and a combined cohort was about 155 million data points that were available. In terms of unique patients, that were about 3 million patients that ultimately filtered down to that had both creatinine and GFR measurements. And what we proposed to do was look at the highest risk cohort of those patients, which are those patients who already have some degree of kidney disease. And we considered that as an eGFR, or estimated glomerular filtration rate, between 15 and 60%. And so that cohort is almost about 3 million patients. So it's a pretty robust data set. And within that data set, we have multiple data points. And for these patients, we have 1.6 million lab values daily. And we're going to add on to this data point and add other data elements that we can use to be able to really allow the algorithms to really do their work. And at this point, I'm going to transition over to Dr. Valafar to talk about what are the tools that he has available and what he's done to be able to really help us answer this.
Is actually data. So what I'd like to do is talk briefly, quickly over some of the applications of AI in healthcare. And from there, I'm going to try to focus on one particular problem that we think parallels very well with this chronic kidney disease and motivate our future work. So AI, artificial intelligence, is really a broader category of the tools that are available. And it goes back as far as the existence of computers. It goes back to about 1950s. Machine learning is a subset of this, is the artificial intelligence that can be applied to machines in order to act more like humans. And there are a variety of categories. You can slice and dice the world of machine learning in different ways. Some examples are expert systems, which are rule-based, whole bunch of if-then statements, supervised versus unsupervised learning, cluster analysis, regression models, classification approaches. and artificial intelligence or deep neural network. This is something that has emerged, has been rejuvenated over the past decade, mostly because of the developments in the deep neural networks. So as the first tool that we are going to use is deep neural networks. And what I'd like to do is highlight some of the examples of artificial intelligence that we have used in neural network in healthcare. One of the simple ones that I can highlight here is we incorporate AI in wearable devices in order to detect human activity, in particular smoking. We have been very successful. We have also utilized artificial intelligence as a way of conducting triage under mass casualty incidents conditions in emergency room where hundreds of people come in a rush, and we rely on artificial intelligence to perform the task of triage. But more closely related to the problem of chronic kidney disease is sickle cell anemia. So I'd like to take a little bit more time in establishing the basis of it and what we have accomplished in this particular domain. So sickle cell anemia is a genetic disorder, is a point mutation along the beta chain of the hemoglobin that causes sickle cell anemia, denoted by HBS. And the single point mutation really kind of incites the self-assembly of hemoglobin as to polymerization of it, and that causes sickling effect of the erythrocytes. Now, so one possible way to mitigate the symptoms of sickle cell anemia is administration of a drug called hydroxyurea. Hydroxyurea increases the expression of fetal hemoglobin in adults, children, and, so of course, at the fetal stage of development. But the point is that it does actually increase the expression of fetal hemoglobin in adults and children, therefore substituting in the percentage of existence for sickle hemoglobin, therefore mitigating the effects of sickle hemoglobin. However, hydroxyurea is a chemotherapeutic agent, and therefore it has some severe side effects. And really, the basic question that we want to address is that can we predict ahead of time who is going to benefit from administration of sickle cell anemia in order to justify the side effects? So as we started this work, my personal expectation was that, well, if people respond positively to a drug, it would appear as an onset that precipitously increases and remains responsive to the drug. What you are seeing here are some examples of different people's response to hydroxyurea measured in the way of the expression of the fetal hemoglobin. So for example, the top left corner, you have example of somebody who immediately started responding as the expression of the fetal hemoglobin increased and kind of maintained that profile until the end. So this drop off, we can assume it's because that's where the data ended. So in the top row, you can see some examples of people who started immediately responding to the drug, although you see in the first row some examples of people who begin, a person who started to, their response decay due to potential rehabilitation to the administration. What is surprising is that you see a variety of profiles of responses. While some people respond, some people don't respond at all. So this would be an example of it would be the person in the bottom row to your hand side, right-hand side. This person through the initial stages of drug administration does not respond, and it is only during month 9, 8, and 9 where their response suddenly kicks in and they become responsive to the drug. There are other people who respond decay. There are people who respond decay in a variety of forms. So it's not that everybody responds in the same way. There are a variety of responses. Some people don't respond. Some people respond immediately, and then from there it tapers off. So really, again, the bigger question is who is going to respond. within this realm of complexity of responses. How well can we do in predicting? So we resorted to artificial neural network. We had 23 genetic clinical measures that we can collect from a given person in a cohort of 120 patients. We followed their fetal hemoglobin for a duration of time, and then we use this information to train and test our neural network. The first measure of response that we used was whether somebody's initial fetal hemoglobin was doubled. Then from there, we refined our definition of positive responder, instead of being doubling effect, to be whether they can surpass a threshold of 15%. And in doing this, we basically constructed the neural network. Sorry, it's not coming across. That basically took 23 of the parameters and had a single output neuron to detect whether 1, meaning a person will be a responder, or 0, a person will not be a responder. Therefore, they should be potentially engaged in the administration of hydroxyurea or not. So under the first categorization of responder, where a person's fetal hemoglobin doubles, among the cohort of participants, we had 100% success in predicting whether a person is going to respond or not. So this was a relatively easier task. For the particular definition of whether a person goes above 15% threshold or not, there we had a success rate of about 85%. So in order to remain agnostic to the definition of who's a responder and not, we decided that we're going to take a different approach. And our approach was not to simply predict whether a person is going to respond or not, but to have a much simpler outcome, but much more difficult to compute. So in here, we engaged in trying to predict where will a person's fetal hemoglobin be 1 month ahead of time. So here, a subject comes in, they receive their administration, the dosage of the hydroxyurea, we extract blood sample and establish their percentage fetal hemoglobin. That information goes into a neural network. We predict what the percentage fetal hemoglobin is going to be next month. And when next month they come in for their routine examination, we compare the actual extracted fetal hemoglobin values to the previously predicted one. So what we are showing here are 3 examples of failures. So these were where we consider we were not successful, although one can, for example, in the bottom one, argue that there's a good correlation, there is a vertical shift in the prediction. However, these are some examples of the successes where we feel we have done fairly decent job of predicting a person's fetal hemoglobin. This is despite the fact that it goes up, down, it's not consistent at all. So a variety of responses are being predicted 1 month ahead of time with fairly good degree of accuracy. We then decided to use this particular technology in order to maybe personalize the administration of fetal hemoglobin to be a little bit more effective. So here is on top row, you see the general way where the drug is administered, that based on height, weight of a patient, you come up with the dosage. In this particular instance, 1,000 milligrams. And this particular person's response is shown in green, and in red is what we had predicted, which is fairly close. But then we can use this predictive tool to play with the potential dosage administration regimen. So instead of maintaining a constant 1,000 milligram, we decided to come up with semi-arbitrarily selected variation of the dosage. Granted, it might be toxic at some points, but that's not the point. The point is that we can come up with alternative administration regimen in order to alter a person's response from what shows to be green into what's predicted to be in red, more stable and steady response to the drug. So I know we are short in time. I'm just going to highlight some of the more important points. In regard to sickle cell anemia, machine learning tool can actually perform a very complex calculations in order to predict not only whether a person is going to be a responder or not, but predict where a person is going to be 1 month ahead of time, and from there be able to personalize, develop an optimized, personalized drug administration regimen. And we are fairly certain that we can apply the same technology with the same degree of success to chronic kidney disease. So at this point, I'd like to take a moment and thank Brendan for all his work, Dr. Roy Matthew for connecting us with this particular problem and helping us, and along the line, a few students who have helped this project, funding from Big Data Health Science Pilot Project and from IMBRI NIH grant that has helped to fill in the gaps. Thank you very much. We hope you have enjoyed this presentation, and please let us know if you have any questions and whether we can help.